Sexual reproduction in flowering plants. Are we not lucky enough that plants reproduce sexually? The myriads of flowers that we enjoy gazing at, the scents and the perfumes that we swoon over, the rich colors that attract us, are all there as an aid to sexual reproduction. Flowers do not exist only for us to be used for our own selfishness. All flowering plants show sexual reproduction. A look at the diversity of structures of the inflorescence, flowers, and floral parts shows an amazing range of adaptations to ensure formation of the end products of sexual reproduction, the fruits and seeds. In this chapter, let us understand the morphology, structure, and the processes of sexual reproduction in flowering plants. Angiosperms. Flower, a fascinating organ of angiosperms. Human beings have had an intimate relationship with flowers since time immemorial. Flowers are objects of aesthetic, ornamental, social, religious, and cultural value. They have always been used as a symbol for conveying important human feelings such as love, affection, happiness, grief, mourning, etc. List at least five flowers of ornamental value that are commonly cultivated at homes and in gardens. Find out the names of the five more flowers that are used in social and cultural celebrations. Have you heard of floriculture? What does it refer to? To a biologist, flowers are morphological and embryological marvels and the site for sexual reproduction. In earlier classes, you have read the various parts of a flower. In figure 1.1, will help you recall the parts of a typical flower. Can you name the two parts in a flower in which the most important units of sexual reproduction develop? Pre-fertilization, structures and events. Much before the actual flower is seen on a plant, the decision that the plant is going to flower has taken place. Several hormonal and structural changes are initiated, which lead to the differentiation and further development of the floral polymodium. Inflorescences are formed, which bear the floral buds and then the flowers. In the flower, the male and female reproductive structures, the androecium and the gynoecium, differentiate and develop. You would recollect that the androecium consists of a whorl of stamens representing the male reproductive organ, and the gynoecium represents the female reproductive organ. Stamen, microsporangium, and pollen grain. In figure 1.2a shows the two parts of a typical stamen, the long and slender stalk called the filament, and the terminal generally bilobed structure called the anther. The proximal end of the filament is attached to the thalamus, or the petal of the flower. The number and length of stamens are variable in flowers of different species. If you were to collect a stamen each from 10 flowers, each from different species, and arrange them on a slide, you'll be able to appreciate the large variation in size seen in nature. Careful observation of each stamen under a dissecting microscope and making neat diagrams would elucidate the range in shape and attachment of anther in different flowers. A typical angiosperm anther is bilobed, with each lobe having two theca. They are diphthous. Often, a longitudinal groove runs lengthwise, separating the theca. Let us understand the various types of tissues and their organization in the transverse section of an anther. The bilobed nature of an anther is very distinct in the transverse section of an anther. The anther is a four-sided tetragonal structure consisting of four microsporangia located at the corners, two in each lobe. The microsporangia develop further and become pollen sacs. They extend longitudinally all through the length of an anther and are packed with pollen grains. Structure of microsporangium. In the transverse section, a typical microsporangium appears nearly circular in outline. It is generally surrounded by four wall layers: the epidermis, endothecium, middle layer, and the tapetum, from the outside to the inside. The outer three wall layers perform the function of protection and help in dehiscence of anther to release the pollen grains. The innermost wall layer is the tapetum. It nourishes the developing pollen grains. Cells of the tapetum possesses dense cytoplasm and generally have more than one nucleus. When the anther is young, a group of compactly arranged homogeneous cells called the sporogenous tissue occupies the center of each microsporangium. Microsporogenesis, the process of creating pollen grains. As the anther develops, the cells of the sporogenous tissue undergo meiotic divisions to form microspore tetrad. As each cell of the sporogenous tissue is capable of giving rise to a microspore tetrad, each one is a potential pollen or microspore mother cell. The process of formation of microspores from a pollen mother cell, PMC, through meiosis is called macrosporogenesis. The microsporogenesis, as they are formed, the microspores, as they are formed, are arranged in a cluster of four cells. The microspore tetrad, as the anthers mature and dehydrate, the microspores dissociate to form each other and develop into pollen grains. Inside each microsporangium, several thousands of microspores or pollen grains are formed that are released with the dehiscence of anther. Pollen grains. The pollen grains represent the male gametophytes. If you touch the opened anther of hibiscus or any other flower, you would find deposition of yellowish powdery pollen grains on your fingers. Sprinkle these grains on a drop of water, take it on a glass side, and observe under a microscope. You will really be amazed at the variety of architecture, sizes, shapes, colors, designs seen on the pollen grains from different species. Pollen grains are generally spherical, measuring about 20 to 50 micrometers in diameter. It has a prominent two-layered wall. The hard outer layer, called the exine, is made up of sporopollenin, which is one of the most resistant organic material known. It can withstand high temperatures and strong acids and alkali. No enzyme that degrades sporopollenin is so far known. Pollen grain exine has prominent apertures called germ pores, where the sporopollenin is absent. Pollen grains are well preserved as fossils because of the presence of sporopollenin. The exine exhibits a fascinating array of patterns and designs. The inner wall of the pollen grain is called the intine. It is a thin and continuous layer made up of cellulose and pectin. The cytoplasm of the pollen grain is surrounded by plasma membrane. When the pollen grain is mature, it contains two cells, one vegetative cell and generative cell. The vegetative cell is bigger than the generative cell, has abundant food reserve, and a large irregularly shaped nucleus. The generative cell is small and floats in the cytoplasm of the vegetative cell. It is spindle shaped with dense cytoplasm and a nucleus. In over 60% of angiosperms, pollen grains are shed at this two-shell stage. In the remaining species, the generative cell divides mitotically to give rise to two male gametes before pollen grains are shed in a three-shell stage. Pollen grains of many species cause severe allergies and bronchial afflictions in some people, often leading to chronic respiratory disorders like asthma, bronchitis. It may be mentioned in that Parthenium or carrot grass that came into India as a contaminant with imported wheat has become ubiquitous in occurrence and causes pollen allergy. Pollen grains are rich in nutrients. It has become a fashion in recent years to use pollen tablets as food supplements in Western countries. A large number of pollen products in the form of tablets and syrups are available in the market. Pollen consumption has been claimed to increase the performance of athletes and racehorses. When they when once they are shed, pollen grains have to land on the stigma before they lose viability. If they have to bring about fertilization, how long do you think the pollen grains retain viability? The period for which pollen grains remain viable is highly variable, and to some extent depends on the prevailing temperature and humidity. In some cereals, such as rice and wheat, and pollen grains lose viability within 30 minutes of their release. And in some members of Rosacea, Leguminosae, Solanaceae, they maintain viability for months. You may have heard of storing semen sperms of many animals, including humans, for artificial insemination. It is possible to store pollen grains of a large number of species for years in liquid nitrogen in the negative 196 degrees Celsius. Such stored pollen can be used for pollen banks similar to seed banks in crop breeding programs. The pistil, megasporangium, ovule, and embryo sac. The gynoecium represents the female reproductive part of the flower. The gynoecium may consist of a single pistil, monocarpellary, or may have more than one pistil, multicarpellary. When there are more than one, the pistils may be fused together in syncarpous manner or maybe in a free apocarpous manner. Each pistil has three parts: the stigma, style, and ovary. The stigma serves as a landing platform for pollen grains. The style is the elongated slender part beneath the stigma. The basal bulbous part of the pistil is called the ovary. Inside the ovary is the ovarian cavity, which has locules. The placenta is located inside the ovary cavity. Recall the definition types of placentation you have studied in the previous chapter in anatomy in class 11. Arising from the placenta are megasporangia, commonly called ovules. The number of ovules in an ovary may be one, like in wheat, paddy, mango, to many, like in papaya, watermelon, and orchids. The megasporangium ovule. Let us familiarize ourselves with the structure of a typical angiosperm ovule. The ovule is a small structure attached to the placenta by means of a stalk called the funicle. The body of the ovule fuses with funicle in the region called hilum. Thus, hilum represents the junction between ovule and funicle. Each ovule has one or two protective envelopes called the integuments. Integuments encircle the nucellus, except at the tip where a small opening called the micropyle is organized. Opposite to the micropylar end is the chalaza, representing the basal part of the ovule. This end is called the chalazal end. Enclosed within the integuments is a mass of cells called the nucellus. Cells of the nucellus have abundant reserve food materials. Located in the nucellus is the embryo sac or female gametophyte. An ovule generally has a single embryo sac formed from a megaspore. Megasporogenesis, the process of formation of female gametophyte. The process of formation of megaspore from the megaspore mother cell is called megasporogenesis. Ovules generally differentiate a single megaspore mother cell MMC in the micropylar region. Of the nucellus. It is a large cell consisting of dense cytoplasm, a prominent nucleus. The MMC undergoes meiotic division. What is the The MMC undergoes meiotic division. What is the importance of the MMC undergoing meiosis? Meiosis results in the production of four megaspores. Female gametophyte. In the majority of flowering plants, one of the megaspores is functional while the other three degenerate. Only the functional megaspore develops into the female gametophyte, embryo sac. This method of embryo sac formation from a single megaspore is termed as monosporic development. What will be the ploidy of the cells of the nucellus? MMC, the functional megaspore, and female gametophyte? Let us study about the formation of the embryo sac in detail. The nucleus of the functional megaspore divides mitotically to form two nuclei which move to the opposite poles, forming the two-nucleate embryo sac. Two more sequential mitotic nuclear divisions result in the formation of the four-nucleate and later the eight-nucleate stages of the embryo sac. It is of interest to note that these mitotic divisions are strictly free nuclear. That is, nuclear divisions are not allowed immediately by cell wall formation. After the eight-nucleate stage, cell walls are laid down, leading to the organization of the typical female gametophyte or embryo sac. Observe the distribution of the cells inside the embryo sac in figure 8.1.8b and c. Six of the eight nuclei are surrounded by cell walls and organized into cells. The remaining two nuclei, called the polar nuclei, are situated below the egg apparatus in the large central cell. There is a characteristic distribution of the cells within the embryo sac. Three cells are grouped together at the micropylar and constitute the egg apparatus. The egg apparatus, in turn, consists of two synergids and one egg cell. The synergids have a special cellular thickenings at the micropylar tip called the filiform apparatus, which play an important role in end and are called antipodals, guiding the pollen tubes into the synergid. Three cells at the chalazal end are called the antipodals. The large central cell, as mentioned earlier, has two polar nuclei. Thus, a typical angiosperm embryo sac at the maturity though 8 nucleate is 7 celled. Pollination. In the preceding sections, you have learned that male and female gametes in flowering plants are produced in the pollen grain and embryo sac respectively. As both types of gametes are non-motile, they have to be brought together for fertilization to occur. How is this achieved? Pollination is the mechanism to achieve this objective. Transfer of pollen grains shed from the anther to the stigma of a pistil is termed as pollination. Flowering plants have developed an amazing area of adaptations to achieve pollination. They make use of external agents to achieve pollination. Can you list the possible external agents? Kinds of pollination. Depending on the source of pollination can be divided into three types. Autogamy. In this type, pollination is achieved within the same flower. Transfer of pollen grains from an anther to the stigma of the same flower. In a normal flower, which opens and exposes the anther and the stigma, complete autogamy is rather rare. Autogamy is such flowers requires synchrony in pollen release and stigma receptivity, and also the anther and the stigma should lie close to each other so that self-pollination can occur. Some plants such as viola, common pansy, oxalis, and commelina produce two types of flowers. Chasmogamous flowers, which is similar to flowers of the other species with exposed anther and stigma, and clistogamous flowers, which do not open at all. In such flowers, the anther and the stigma lie close to each other when the anthers dehiscence in the flower buds. Pollen grains come in contact with the stigma to effect pollination. Thus, clistogamous flowers are invariably autogamous as there is no chance of cross-pollination landing on the stigma. Clistogamous flowers produce a short seed set even in the absence of pollinators. Do you think that clistigamy is advantageous or disadvantageous to the plant? And why? Gatogamy. Transfer pollen grains from the anther to the stigma of another flower in the same plant. Although gatogamy is functionally cross-pollination involving a pollinating agent, genetically it is similar to autogamy since the pollen grains come from the same plant. Xenogamy. Transfer of pollen grain from anther to the stigma of the different plant. This is the only type of pollination which during pollination brings genetically different types of pollen grains to the stigma. Agents of pollination. Plants use two abiotic wind and water and one abiotic animals agents to achieve pollination. Majority of plants use biotic agents for pollination. Only a small portion of plants use abiotic agents. Pollen grains coming in contact with the stigma is a chance factor in both ways.